basically, yeah. Let's see.
There we go.
Okay,
yep.
We're live on Facebook, and
all right, it is 3 o'clock, so we will get started. Welcome everybody to this week's seminar. I will start by reading the land acknowledgement. We respectfully acknowledge that the territory and lands on which Memorial University campuses are situated are in the traditional territories of diverse Indigenous groups, and we acknowledge the histories and cultures of the Beothuk and the island of Newfoundland as the ancestral homelands of the Mi'kmaq and Beothuk. We would also like to recognize the Inuit of Nunavut and Nunatuhavut and Innu of Nitassinan and their ancestors as the original people of Labrador. We strive for respectful relationships with all peoples of this province as we search for collective healing and true reconciliation and honor this beautiful land together. We finally encourage everyone to reflect on these lands from where you are located and the Indigenous peoples for whom these lands are traditional territory. Thank you everyone for joining us. I would like to introduce our speaker this week, Dr. Kirsten Meyer-Kaiser, who's from Woods Hole Oceanographic Institute, and will be speaking today about recruitment of deep sea invertebrates. Go ahead whenever you're ready, Kirsten.
Great, thank you so much for that, Carly. I'm very excited to speak to this group today, and I'm hoping that we can have a good presentation and maybe some good questions after as well. Since I'm talking about settlement and recruitment, I thought it would be best to start out by defining those terms. I work on, very specific marine invertebrate larvae, and these might not be intuitive terms to other types of biologists. So if you picture a habitat on the seafloor colonized by invertebrates, such as this one. For example, you have some sponges here, there are brittle stars, there are bryozoans on this rock. Those adults are going to produce larvae. Those are the early life history stages of those invertebrate species. And I've added some examples here. This is a cyprid. It grows up to be a barnacle. This is actually a juvenile brittle star. Sometimes they'll metamorphose in the water column, so you can actually catch juveniles. But then this is a larval bivalve. We call it a veliger. So that'll grow up to be a clam or a mussel. Larvae disperse in the water column. They are just carried around by ocean currents, taken to new habitats where they metamorphose in order to lead their adult life. And so the process by which they come out of the water column, they descend down to the seafloor and find a place to attach and metamorphose. We call that settlement. It's everything that happens from when they're coming out of the water column to when sometimes they'll scooch around on the substratum until they find the optimal little microhabitat with the correct flow for their feeding, and then they'll attach and they'll metamorphose. That whole process we call settlement. But then there's recruitment, which is defined operationally as surviving until the researcher comes back. And so there's actually a pretty big difference in some case between those, driven by what we call post-settlement mortality. This is if there's an environmental perturbation, if there's predation, if there's competition, you can have a really steep decline in the number of settlers. So there's some things that a researcher is not able to immediately observe in terms of those settlement processes that we can only see as recruitment. So that's the difference between settlement and recruitment. I work on both. So a lot of my research takes place in the Arctic, specifically this little area. This is Svalbard. It's an archipelago halfway between northern Norway and the North Pole. It's centered at around 78 degrees north. And being in the Eastern Arctic, Svalbard is actually uniquely warm for its high latitude. So this is a satellite image of sea ice cover in the Arctic. And just to orient you, Svalbard is right there. And you can find Newfoundland, obviously down here, surrounded by much more sea ice at lower latitude. And you've got ice all the way across northern Canada at lower latitude than you find at 78 degrees north in Svalbard. This is driven by oceanographic circulation. You have this current system that's bringing warm saline water that ultimately originates in the tropics through the North Atlantic and up into the Eastern Arctic. As it goes past the eastern coast of North America, we call it the Gulf Stream. It veers offshore right around Cape Hatteras, North Carolina, and goes across the Atlantic as the Gulf Stream extension. As it goes past Europe, we call it the North Atlantic Current. As it goes past Norway, we call it the Norwegian Current. And eventually it makes its way, one branch goes into the Barents Sea and one branch goes into the Fram Strait, which is this passage here between Svalbard and Greenland. And we call that the West Spitsbergen Current. So the West Spitsbergen Current is responsible for bringing very warm, very saline water, ultimately originating in the tropics, up through the Eastern Arctic and warming that western area of Svalbard. So it's very unique for its high latitude. I'm a benthic ecologist, which means that I study all sorts of organisms that live on the seafloor, things like this. This is a seafloor photograph that we took just north of Svalbard at about 300 meters depth on the continental shelf there. So this is just some examples of sessile invertebrates. You've got some bryozoans here, like this white branching form. There are some tube worms, like these little white things. This isn't sessile per se, but these yellow flower-like things, those are feather stars, crinoids, and they come to rest on a lot of the substrata. So we see them frequently on rocks around my study sites. Another example, this is at greater depth. This is at about 2,500 meters west of Svalbard in that deep Fram Strait area. You'll see the white flowery things here. That's actually a glass sponge. Its species name is Calophacus arcticus. It's very common. You can see that there's this white branching form here. That's a different kind of sponge. It's actually a carnivorous sponge, which I think is very cool, and I will be happy to talk about carnivorous sponges later if you're interested. We also see there's a pink anemone here, and there's a red shrimp hanging out on a dead sponge right there. So we get a lot of organisms on those rocks on the continental slope west of Svalbard. The rocks themselves are called drop stones, and they actually originate from land through this very unique process. First step is they're frozen into glaciers. Each of the fjords in Western Svalbard has a glacier at the head, and they look like this. This is from Kongsfjorden, one of the ones in Western Svalbard. And you'll notice in amongst this very blue ice, there's this brown patch right here. That's going to be terrestrial debris. It's going to have some little pebbles, maybe some larger rocks that get frozen into that glacier. And then when the glacier melts at the front edge, when it reaches the ocean, it calves off icebergs, which then carry those rocks out to sea. And when the iceberg melts, the rocks fall to the ocean floor, hence their name, drop stones. Drop stones are seen all around the Arctic in those glacier-influenced areas. And so just as one more example of benthic communities in my Svalbard study area, this is a rather steep cliff. It's a rocky reef at about 1,800 meters, again, west of Svalbard. And again, you can see there's all sorts of sponges, these white tennis ball sponges. Those are a very common species called Tentorium semisuperites. You've got that white branching form there again. That's that carnivorous sponge, Cladorhiza. And again, you have a little red shrimp. So looking at communities like this, my question is, how did they get there? How was that rocky cliff or that drop stone colonized and through the processes of settlement and recruitment? And that's what I'm trying to investigate. So when you have, that brand new substratum, recruitment is that process that turns it into a robust community. And we can simulate that process through experiments by putting out experimental substrata. We call these fouling panels. I spent a lot of time over the past 5 years putting out fouling panels in Svalbard waters. And when we recover them, we can see what has recruited, what would be the first colonists in that community. So the first study that I want to tell you about today is taking place in this area called the House Garden. This is an LTER, a long-term ecological research observatory. This is maintained by the Alfred Wegener Institute. I've had the opportunity to collaborate with Avi over the course of the last 10 years. I used to live in Germany, and then when I returned to the US for my graduate studies and now into my own research career, I've continued to work with them. And so the House Garden is just a series of stations that are sampled in the same way every year. So we get a nice long time series. But having that access to these high latitude environments gives us a really unique opportunity to also run experiments building off of that long-term observatory. So using the central station in the House Garden, that's marked here by this orange star, it's at 2,500 meters depth, 79 degrees north, 4 degrees east. I undertook a really unique experiment, and more properly said, my Avi colleagues began this really unique experiment in 1999. Obviously, I was not present for the beginning of that. I was in the 4th grade, but this metal frame was put down by Michael Klages. He was at the time the director of the Avi Deep Sea Ecology and Technology Group, with the goal of understanding on a long-term basis how those hard-bottom communities on the continental slope west of Svalbard are formed through those processes of settlement and recruitment. We were only able to observe recruitment, obviously, because the researchers had to come back, and so there was that post-settlement mortality that happened. But we did come back, and in 2003, my Avi colleagues were able to visit this metal frame that they had deployed with an ROV, a remotely operated vehicle, and there was really not much on it. They actually managed, using the ROV's manipulator arm, to cut off a couple of those panels, take them back to the lab and look at them, and there were a couple of foraminiferans on the panels, but no animals to speak of. In 2011, this was actually my first cruise with the Avi. We again visited this frame with an ROV to see what had recruited to it, and there were some hydroids. You could see them on the panels. We didn't collect any that year because it seemed still like the colonization was pretty low. So then we left it until 2017, and after 18 years on the seafloor, finally brought the metal frame with all of its panels back to the surface, and I had the opportunity to lead the analysis of those results. And we found 13 species. So just to give you in a more visual way what that looked like, this is a frame grab from the ROV video from 2003, and you can see there's really not much there. Maybe a juvenile shrimp hanging out over here, but no sessile colonists to speak of. 2011, you can see the hydroids on the panels. The streaks are called stolons. Those are parts of the hydroid colony that grow flat along the substratum. And in 2017, again, you can't really see much. You can tell that there's hydroids, and then those white tubes that you see, those are some tube worms. But we thought that we might be missing some things with just looking at the ROV video, which is why we decided to pull it up. And having those panels physically in the lab, able to go under the microscope, revealed much more than the ROV video. So I'm glad that we did pull it up. So we've got 4 morphotypes up here, panels A through D. Those are foraminiferans. Most of them are unidentified, but this panel C, this is a very common suspensions feeding foram in the Arctic. It's called Civicidites willardsdorfi. Some of you may have heard of that before. Panel E, I eventually figured out that's a glass sponge. It's probably the same species as those white flower-like ones that you saw earlier. They were all over the backside of the brick panels. They really liked to recruit in the microhabitats. So these like little divots. These were brick pavers that you might use in your garden. And so they had these triangular ridges, and down in that depressed area was where most of those glass sponges settled. This is another very common species of sponge from the House Garden. This is that carnivorous sponge. which I thought was really fascinating to find. It kind of looks like a cactus to me. And so those spikes function as a type of Velcro. It feeds by having copepods get stuck on the spikes, and then the sponge digests it once the copepod is stuck there. It's really cool. This is that hydroid that we were seeing, Helicophonia arctica. This is a soft coral, which again is really common in the house garden area. Anemone, super common. There's that tube worm, and this one is actually kind of sticking up off the panel. You can see the shadow of its tube there. And then this is a crinoid. This is actually a juvenile feather star, which I'll speak about more in a minute. So finding each of those species was very exciting, and even more so because I could match what we were seeing as the recruits to common adults in the area. But I do have to mention that they're not necessarily in the same proportion that you would expect. For example, this tube worm was in very high numbers on our panels. We got something like 300 individuals on the long-term experiment. But if you look at any of the drop stones in the area, there's maybe two tube worms. And we've even trawled up some of the stones and just very, very low numbers of tube worms. So there was a really strong recruitment, very much higher numbers of recruits than we ever see in the mature communities. The same thing is true for this feather star and for a couple of the forams as well. So different proportions of organisms settling and recruiting to this experiment. The other thing that we were able to do was compare the community of recruits based on physical characteristics of the experiment. So we knew what orientation the metal frame had been deployed in, and obviously it had sat in the same spot for 18 years. Comparing that to the predominant bottom current, I've got annual averages up on the top here, and you can see that the current is very consistent. It is between 7 and 8 centimeters per second to the northwest all the time. So I was able to differentiate between panels that were facing different angles to the predominant bottom current. And of course, we also had two different types of panels. I don't know if you noticed from the photos, we had plastic and brick. And then we also had them at 3 different heights. So there were some panels that were right on the bottom in contact with the seafloor, some that were about 60 centimeters off the bottom, and some that were about 90 centimeters off the bottom. And we were able to compare those heights. Interestingly, we also had a radar reflector that was on a staff about 2 meters off the seafloor, so much higher than any of the panels. And we found some recruits on that radar reflector as well. This is that carnivorous sponge, which obviously has grown to a much larger size than anything we saw on the panels. And this is also a species of hydroid, this pink flower-like thing at the very top. That hydroid and the sponge and most of the other organisms that we found on the panels make their living by feeding on things out of the water column, either small particulates, they're suspension feeders, or in the case of that carnivorous sponge, copepods that they're capturing as a predator. And so I expected that we were going to have much higher densities of recruits higher off of the seafloor because they need to be exposed to as fast of a current as possible in the benthic boundary layer, wanting to have that advantage in suspension feeding. So I compared the recruits that we found based on the characteristics. We did find a significant difference between panels made out of those 2 different materials, the plastic and the brick. That was significant on the community level. As I've mentioned, there were some species that were more abundant on the brick panels, for example, that glass sponge that liked to be in the microhabitats. The brick was a much better representation of, or a much better analog for drop stones and natural hard substrata that you'll find in the area. So I considered that to be more of an indication of what the natural recruitment might look like, whereas the plastic was more indicative of some anthropogenic substrata. There's actually a surprising amount of litter in the Fram Strait because it's north of Europe, and so it's carried on ocean currents into the Eastern Arctic. And so we can conclude that those two substrata do not necessarily get the same recruitment. In terms of the height off bottom, we had on the bottom 60 centimeters off bottom and then 90 centimeters off. And again, we had a significant community level difference. There were some species specific patterns that I'll show you in a minute that were actually a little surprising. It wasn't necessarily the case that everything was more abundant higher off the bottom. But then for panels at different orientations to the bottom current, we did not have a significant community level difference, but there was one species that was more abundant. I'll show that in a second. So these species specific patterns were really interesting to me. This hydroid, for example, was much more abundant, higher off the bottom at 90 centimeters above the seafloor, which I expect has to do with the advantage that it gains in suspension feeding. It wanted to be higher up or had an easier time surviving higher up. Because again, remember we're looking at recruitment patterns here, which include that survival component. Our carnivorous sponge didn't have any significant differences across the different heights. But this tube worm, this actually really surprised me. It was actually more abundant on the bottom, so on panels in contact with the seafloor. It's also a suspension feeder, so I was really surprised to find that. And the one reason that I can think of maybe is if their larvae are demersal, meaning that they hang out right near the seafloor, maybe that circulid, that tube worm would only have encountered or would have encountered the panels in contact with the seafloor at higher density. I'm not quite sure, I can only speculate about that, but it was really interesting to me that it was so much more abundant lower down. And then I've mentioned the angle to the bottom current. The glass sponge actually was about 3 times more abundant on panels that were in line with the bottom current. And I suspect that the reason for this again comes down to suspension feeding, because most of the organisms that we found on these recruitment panels had some sort of flexible element to their morphology. You know, the tube worm has its filtering crown that can sway in the current, or the carnivorous sponge or the soft coral might be able to change their body shape. Whereas this glass sponge is rather rigid, it doesn't have that opportunity to change shape. So maybe there was higher settlement or there was higher post-settlement survival, leading to higher recruitment on those panels in line with the bottom currents, so that it had an advantage in suspension feeding. Really interesting species-specific recruitment patterns here. And the last species that I want to talk about is this feather star. This is actually a little bit of an aside, but you'll notice that we found a lot of this species of feather star, this crinoid, not just as recruits on the panels and on the frame, but we also found some adults resting on that frame. And I've shown the adults with the yellow arrows here, the ones that don't have a conspicuous stalk. So I'm not sure how much this group knows about crinoid development, but the feather stars that swim in the water column as adults actually have early life history stages that are stalked. And so they're attached to a surface. And eventually the crown breaks away and swims off into the water column and does its thing. So we were able to describe the ontogenetic development and actually the process of metamorphosis in this species. It's called Poliometra prolixa, and it's a common eurybathic Arctic crinoid, but we had such a high number of samples, such a high number of individuals that recruited onto our panels that we were actually able to observe the different stages. So it starts with this diamond-shaped head. The stage is called the cystidian. And so it's just a very simple stalked form with that diamond-shaped head. The ossicles close over the mouth and then undergo this transitional process in which they're reshaped and eventually form a head with some simple arms. And that form with the arms is called the pentacrinoid stage. Those arms eventually bifurcate and grow into the swimming arms of the adult crinoid that we know. And so it was actually very exciting to be able to describe that metamorphosis for this species that had never had its development fully described before. And so the insights that we gained from this long-term recruitment experiment were actually pretty powerful for understanding how communities form in the Arctic deep sea. We can first say that Arctic benthic recruitment is pretty stochastic and patchy, because we had really high abundances of some species that were not necessarily very abundant in the background communities, but then complete absences of other things, so that there's an element of stochasticity and patchiness to that recruitment. Again, I said we had high numbers of individuals from just a few species, and we can also say rather confidently that these communities take decades to develop. We had this experiment underwater for 18 years, and we caught 13 species. Just as a reference, in the background community, there's about 65 hard-bottom morphotypes. So we only got a very small proportion of what is present in the study area. And so we suspect that, I'm not sure exactly how long this would have to stay on the seafloor to fully capture that diversity and start to look like the background communities, but much longer than 18 years. All right, at this point, I'm going to switch gears a little bit, because that covers a recent experiment that has provided some insights for deep sea recruitment. And now I want to tackle the question of settlement, looking at those dynamics for things that are attaching and metamorphosing, but then potentially dying before the researcher has a chance to observe them. So recently, I have been collaborating with some engineers at my home institution, Hui, in order to develop a camera system that gets at those settlement dynamics. We're calling it CATAIN, which stands for Camera to Analyze Invertebrates. And yes, CATAIN is meant to be a play on Catan, as in Settlers of Catan, because it photographs settlement. So, bit of nerd humor there. The camera system basically works like this. It's got a machine vision camera with a macro lens on the inside of an underwater housing. Of course, it needs a battery and some simple computers in order to power that. But the camera is focused on the end cap of the housing, which is clear. And so we're using the end cap as the fouling panel itself. And so things will settle on that end cap. They're illuminated with a ring of LED lights that goes circumferentially around the end cap of the housing, so that that reduces glare. And then at regular time intervals, we're able to light it up, record an image of what is settling on the end cap, and then be able to tell what has arrived, what has died, and get at those settlement dynamics with pretty high temporal resolution. So this is what the inside of CATAIN looks like in real life. These metal bars on the sides that provide support, that's called the chassis. And this big blue thing down here is just a cord that's wound up. It's actually a pretty simple system. The guts are all just here in the middle. That's a little Raspberry Pi computer with a Sleepy Pi shield. Sleepy Pi is a type of computer specifically designed for going into a low power mode and then waking up at set intervals. So it wakes up, powers up the camera, takes an image, and then goes back to sleep. This helps us conserve our battery life for long deployments. And then we've got that macro lens attached to a machine vision camera, and you can see that it's focused on the end cap, which is this clear piece here at the bottom, and the LED lights are under this black strip going around the edge. That's it. It's actually quite a simple system. But we were able to put this under the Hui pier at about 16 meters depth for its first test deployments recently, which was very exciting to actually see that it works and get some results. My goal eventually is to use this in much more remote environments, put it in the Arctic deep sea. But for test deployments, we had to stay with an easy environment at 16 meters. So we attached it to this PVC holster. This is basically a rectangle of plastic filled with concrete. And then we've got these circular holsters that are holding the camera in place. Lower that over the side of the dock with a crane, and then we were able to move it into position on the seafloor by scuba diving. And you can see here in this underwater image that we've got a couple of settlers on the end cap. This is from last fall. There are 2 white dots there, those are barnacles. And then we've also got some mobile predators that have come onto the system. These are some predatory snails here that are on the system. And so if I can show you some example images, this is from very early on in our deployments. This is an image, again, through the underside of that housing, and you can see the lighting from the LEDs around the side. This is one of those snails crawling its way across the lens. And so you can see there's the muscular foot here, there's the shell, which is a little out of focus because it's outside of the focal plane. And I'm not sure if that little structure there might be a radula, but we can definitely tell that that is a snail crawling across the end cap of our Catene system. This is an image from last July, a little later in the deployment. You can see that there's more stuff on the end cap. You can tell what some of it is. So this little white guy, that's barnacle, balanus. And then this flowery thing here, that is a colonial ascidian, Botuloides violaceus, which is actually, interestingly, invasive. But it's so common in fowling communities in New England that it may as well have been there forever. So I was excited to see that as a settler on our end cap, one of the very common species in the area. So things are obviously going to grow over time. And this is another image from, again, later in the deployment. At this point, the camera had been underwater for about a month. And you can see there's several different species living here. So this thing that I've marked with an A, that's a tube worm. It's a sand grain tube with a polychaete inside. Thing marked with a B here, this is an arborescent bryozoan called Bugula. So most of its branches are out of focus because it's growing up and away from the camera, but it's got an attachment point on the end cap. This is Balanus, again, another barnacle. This white colony here, marked D, that is an encrusting bryozoan, Cryptosula. And then we have another one of those snails. This is Asteris lunata on our end cap. And so in order to enable the image analysis, we decided we were going to crop off some of the edge areas so that we focus on what's most in focus, not warped in the middle of the camera. And we're still able to get a really good area. So my grad student actually has gone through and counted and marked all of the organisms on that set of test images that we have from last July. And one of the really cool things that we found in that first test deployment is that we can actually see post-settlement mortality happening. We can finally observe those dynamics that have eluded researchers, because previously we were only able to study recruitment. So this is, again, a colonial ascidian, Botuloides. You can see the colony here, and then in the next image, it's gone, and there are just some scraps of tissue left behind. So whether it was predation or some sort of environmental perturbation, that ascidian has died and suffered post-settlement mortality and disappeared from the camera. And you can see that happening. So for the image analysis pipeline, which again, as I mentioned, my grad student has been working on, we first start with just downloading the images, and we adjust the brightness to make it easier to see and resolve everything in the image, crop off those edges. And we found this tool called Label Me it works in Python, and you can actually draw polygons around each of those organisms in order to track not just their presence, but also their size over time. So we're able to measure growth rates as well. Those data are exported in a specific file format, JSON, and then collated into a spreadsheet so that we eventually get the size and the location of every individual on our end cap in each of those images. And so this is what we found. We're actually able to measure settlement rate over time. We defined settlement as being not present in one image and then being present in the following image. That organism has then arrived some point in between the images, and we recorded images every 4 hours in this test deployment. So then we can find the settlement rate per day for each of these different species. And so you can see our friendly barnacles, here shown in pink, have just a relatively low and constant settlement rate, arriving about 1 or 2 individuals per day. On the other hand, our bryozoan, Cryptosula, has a peak in settlements in the middle of our month-long deployment and then declines toward the end. So maybe there was some sort of reproductive event that released a lot of larvae into the water column, and maybe many of them settled at the same time. Not quite sure, but we definitely see that increase in settlement in the middle of the month. And then Hydroides dianthus, this is a tube worm that appeared on our end cap. So it's got nothing is settling, nothing is settling. And then finally, towards the end of our month-long deployment, it's got some settlement there, resulting in a peak towards the end. So we can actually see the timing of settlement within a deployment for each of these different species, which is pretty exciting. And as I mentioned before, we're able to measure the growth rate. So again, we've got that colonial Ascidian. I've shown 2 different individuals here, but just a trend line for one of them. They have kind of a linear growth, increasing in size, here measured in square millimeters, in kind of a linear fashion. And then where that line ends, that's when the mortality happened and those individuals disappeared. We were also able to measure growth again for that barnacle and for our friendly bryozoan, but those both take more of an exponential type of growth pattern, which was, again, really cool to see. They're increasing in diameter. That leads to that exponential curve. And I've shown one trend line per species here, just one example individual. And so right now, we're still developing and using this system in Woods Hole, but we know that it can effectively measure settlement rates. It can monitor post-settlement mortality. It can be used for growth rates. I'm hoping eventually that we can also use it for monitoring, studying overgrowth competition, because you might be able to see those 2 organisms, one overgrowing the other, observing from the underside. It can be used for animal movements. We've got those mobile predators, those snails that are crawling over the end cap. We're keeping track of those snails in all of our deployments now in order to see if there's any seasonal or daily patterns in their movement. And it could eventually be modified for experiments. So, for example, if you wanted to seed the end cap with individuals of a certain species and then watch their growth or their post-settlement mortality over time. And as I've mentioned, my goal is to eventually deploy this in remote environments such as Arctic and Antarctic, stick it on deep sea moorings, put it in remote locations, anywhere that a researcher cannot access on a daily basis. This would be a really effective way for collecting those data, studying settlement in a way that we never have been able to before. And so with that, I think I will conclude, and I will be happy to answer any questions that you might have about either study. So thank you very much.
Thank you so much, Kirsten. If anybody has any questions, you can type it in the chat box or just unmute yourself. I actually have a question, Kirsten, so I'll start off.
Sure.
Can you tell me about any of the predators that the invertebrate settlers that are sessile have on your plates for reasons that they might be gone after a certain amount of time? Or is there any way to know why they're there one day and gone the next?
Yes, so in the case of the Ascidians, I'm wondering whether it was some sort of a temperature shock because the Ascidians disappeared at the same time. I mean, it's also possible that we could have just gotten a predator coming through and eating everything. So the whelks that we see, there's some predatory snails that are crawling over the end cap that I know eat barnacles, and so they'll drill into those shells. So we're trying to actually see if we can tell whether a barnacle is alive or dead from the underside by looking at how opaque or transparent it is. But having those predators come through and eat what is there is one of the things driving mortality.
Gotcha. Okay, thank you. We have a question from the chat box. Why are the growth curves for different species different? And what influences whether the growth curves are linear or exponential?
That is an excellent question, and I don't know. So the exponential is actually more of what I expected, because you have essentially a circular footprint that is then increasing in diameter. So the bigger it gets, the more the increase the next time, you know, it grows. And so I would expect that to result in an exponential curve. I was actually really surprised to see that the Ascidian had more of a linear growth curve, because I would not have expected that for something with a circular footprint. They're also adding zooids as they grow, as opposed to a barnacle that's just a single individual increasing in size. Maybe that has something to do with it. Someone who knows more about invertebrate growth or things controlling growth rates might be able to make better comments on that. But I was very interested that they were different. Looks like another question in the chat. Yeah, how did we deploy the settlement frame in Svalbard? Was it free fall? No, it was not free fall. We actually lowered it down, and I say we very loosely because this was 1999, but I know because it was the reverse of how we recovered it. But it was lowered down on the winch cable, and so it, you know, hit the seafloor very softly, controlled from that winch cable, and then the ROV was deployed to then go and pull the quick release underwater and disconnect the winch cable. So it was kind of a delicate operation with, you know, the winch cable and the ROV tether having two lines in the water at 2,500 meters. They were able to do it quite well and just have that lowered and then released once it was already on the seafloor. Great. Any other questions?
Hi, Kristen. Great talk. Thank you. Very interesting. I have been working with some baited camera footage, so I can, I guess, somewhat relate to the trials and tribulations of camera data and trying to translate that into something meaningful. I was impressed with the setup in terms of its simplicity, but also its sophistication. And so your camera setup, you said it was deployed for about a month. Like, is that the maximum timeline for how long it can be deployed?
Yeah, so right now our limiting factor is the battery life.
Okay, that was going to be my follow-up. Sorry.
Sorry?
Yeah. No, I was just going to say, is the limitation a storage issue or is it a battery issue?
Right now, it's a battery issue, which could easily be solved if we added a second battery. There's plenty of space inside the housing for that. We just haven't done it yet. So right now, our battery will support 400 cycles. So that's just depending on how frequently you want to be taking your images. And we've decided to record every 4 hours so that we have some possibility eventually of detecting tidal patterns, which would potentially be on a 12 or 24 scale. So we wanted to have higher resolution than that. If we, wanted to put it out for a year, we could just set it to 24-hour cycles and then be able to get 400 days. So that was one part of your question, is what was the limiting factor? And what was the other part of your question?
Oh, just how, basically how long can you deploy the unit for? And I guess it's just dependent on what how you program it. Exactly.
So right now we've been doing 2-month deployments. The other limiting factor that could come up is getting the end cap 100% fouled. And because then you can't see anything else that's arriving. And so last summer, after about a month in the water during the highest settlement period of our year, it was actually pretty darn close to 100% fouling. But if it weren't for the battery issue, honestly, right now, I could have left it out for 6 months. There's like 2 barnacles arriving. So we've been putting it out for consecutive 2-month deployments in hopes of getting a year-long record right now.
Awesome. Sorry, I just got my camera working now. I had an additional question about, have you done any ground truthing on your species identification for the unit? Obviously, when you're looking at panels from your deep sea work in Svalbard, you're looking, top down on top of the animal